Hello everyone, I am Dr. Nilanjana Bhattacharya Nath from Swami Vivekananda Institute of Modern Science. I had already discussed about the passive transport in my previous video, and in this segment I would like to talk about the another transport system, that is active transport. So let's start with a brief introduction. Active transport is the process of transferring substances into, out of and between cells using energy, usually in the form of ATP. If a substance must move into the cell against its concentration gradient, that is, if the concentration of the substance inside the cell must be greater than its concentration in the extracellular fluid, the cell must use energy to move the substance. Some active transport mechanisms move small molecular weight material such as ions through the membrane. In addition to moving small ions and molecules through the membrane, cells also need to remove and take in larger molecules and particles. Some cells are even capable of engulfing entire unicellular microorganisms. So, depending on the overall process, active transport system includes number 1, primary active transport, and number 2, secondary active transport. Next, electrochemical gradient. Before starting the detailed discussion of different types of active transport, I would like to discuss the basic concept of electrochemical gradient. As I have discussed simple concentration gradient in my previous video, in which a substance is found in different concentrations over a region of space or on opposite sides of a membrane. But in living systems, gradients are more complex because atoms and molecules can form ions and carry positive or negative electrical charges. There may be an electrical gradient or difference in charge across a plasma membrane. In fact, living cells typically have a membrane potential, an electrical difference or voltage difference across their cell membrane. As an example of how the membrane potential can affect ion movement, let's look at sodium and potassium ions. In general, the inside of a cell has a higher concentration of potassium ion and a lower concentration of sodium ion than the extracellular fluid around it. Thus, in a living cell, the concentration gradient and electrochemical gradient of sodium ion promotes diffusion of the ion into the cell, and the electrical gradient of sodium ion, that is a positive ion, tends to drive it inward to the negatively charged interior. The situation is more complex for potassium. The electrical gradient of potassium ion promotes diffusion of the ion into the cell, but the concentration gradient of potassium ion promotes diffusion out of the cell. This combination of concentration gradient and voltage that affects an ion's movement is called electrochemical gradient. Next, primary active transport. Primary active transport moves ions across a membrane and creates a difference in charge across that particular membrane. This type of active transport system uses ATP to move a substance, such as an ion, into the cell, and often at the same time a second substance is moved out of the cell. The sodium-potassium pump, an important pump in animal cells, expends energy to move potassium ions into the cell and a different number of sodium ions out of the cell. The action of this pump results in a concentration and charge difference across the membrane. Next, secondary active transport. Secondary active transport describes the movement of material using the energy of the electrochemical gradient created by the primary active transport system. Other substances such as amino acids and glucose can be brought into the cell through membrane channels. For instance, a carrier protein lets sodium ions move down their gradient but simultaneously brings a glucose molecule up its gradient and into the cell. The carrier protein uses the energy of the sodium gradient to drive the transport of glucose molecules. Depending on the nature of the molecular transport across the membrane, we can divide the active transport in two categories. In secondary active transport, the two molecules being transported may move either in the same direction, for example, both into the cell, or in opposite direction, that is, one into and one out of the cell. When they move in the same direction, the protein that transports them is called symporter, while if they move in opposite directions, the protein is called an antiporter. In the third type of active transport, large items or large amounts of extracellular fluid may be taken into a cell through the process of endocytosis. In endocytosis, the cell uses proteins in its membrane to fold the membrane into the shape of a pocket. This pocket forms around the contents to be taken into the cell. In receptor-mediated endocytosis, a cell's receptor may recognize a specific molecule that the cell wants to take in and form a vesicle around the area where it recognizes the molecule. Exocytosis is the opposite of endocytosis. In exocytosis, the cell creates a vesicle to enclose something inside the cell for the purpose of moving it outside of the cell across the membrane. So, from the overall discussion, we can conclude that active transport is the term reserved for transport processes that result in the movement of a solute uphill or against its natural direction. For the case of a neutral solute, this resolves into movement against a concentration difference. For a charged solute, it is movement against the combination of concentration as well as the electrical potential differences. This system therefore requires the investment of energy. So, the basic difference in active transport versus passive transport is the energy requirement. Passive transport occurs naturally, but active transport does not occur naturally. And passive transport does not require any kind of energy, but active transport only occurs in the presence of active forces or energy. Thank you.